Good morning, everyone. Thank you very much for participating in our webinar. My name is Professor Luis Avila. I will be hosting the webinar today. I am professor at the Weed Science Research Group at Federal University of Pelotas. Pelotas is a city in the southern Brazil. In our research group, we are 6 weed scientists, and we perform research in a wide variety of issues in weed science. You are welcome to visit our website. We have information on our group there. One of our duty as a scientist and a professor is providing education to young scientists. For this reason, we assembled this webinar series to train the next generation of weed science. Before we start, I would like to mention a couple of information. During our presentation, you can ask any question you want in the question and answer button in the bottom of your screen. You can ask the question during the presentation, and at the end, we're gonna select the question, and we're gonna Dr. Napier will answer the question at the end. Everyone who is registered to this webinar doesn't need to register to the next one, because we have only one registration. You're going to receive all the invitation during the next webinar, so one registration is enough. Well, again, thank you very much all for your attendance to our webinar. Let me introduce our next speaker, Professor Richard Napier. Dr. Napier is professor at the University of Warwick and is the world expert on Auxin Receptor and Auxin Herbicide mode of action. Today, he will be speaking about Auxin Herbicide mode of action and resistance. Dr. Napier, thank you very much for accepting our invitation. I'm very glad to see you and very glad to have you in our webinar series.
Thank you very much. Let's go to share screen. Okay, can everyone see that?
Yes.
Yep, okay, good. Thank you very much, everyone, and good morning. Good morning to those in Brazil. Good afternoon to those who are a little bit further east than that. And as Louis just explained, I work on the mechanism of action of auxins as hormones in nature, in plants, but of course that also translates across to how they work as herbicides when we apply them as synthetic auxins in the field. And I'll be going through some of our work today. And what I'm doing is I'm looking at right at the very start of the whole process of what happens when an auxin.
Richard?
Yeah?
It's not moving the presentation.
Okay, I haven't gone forward yet, so.
All right.
Is that going?
No.
Ah. Okay, I'm going to go back, start again. Apologies, everyone. This worked before. How are we doing now?
Yeah, that's okay. No.
Okay. But you heard what I said before.
Yes, we did.
Yeah, okay, I won't go over that bit again. Okay. So my research is based on the mechanism of action, but let's just all start because I know we start at the base of weed science, and of course the great interest in auxins as commercial prospects is as their use as selective herbicides. And whether you're looking after turf grass or your garden or or cereals or rice, you'll be using them to kill, by and large, the broadleaf weeds. The next slides just introduce the value of herbicides, again, as a basis for what I'm going to say. And I accept that the figures I've got on here are probably a few years out of date now, but we'll all accept that the global herbicide market is vast, enormous, valuable. And although auxins clearly are not, by volume or weight, the most abundantly used herbicides, they contribute a very large part of the arsenal that we have for controlling weeds to ensure our food security. So here are some figures from a little bit while ago about, we get down to the auxins now. So of course, one of the oldest and one of the most widely used still is 2,4-D. And back in 2011, the American figures in pounds, or the international units of kilograms and tons there, show that there are vast quantities of 2,4-D being used in the US alone, and of course elsewhere around the world. And 2,4-D is used in 10% or more of the planted area of a huge range of crops to help maintain the security of their yield. And I'm not sure how widely known the Henry Ford Foundation prizes are, but in 2004, 2,4-D was one of the 75 most important innovations of modern times, according to that foundation. So that just goes to show the importance of auxins as commercial prospects. And of course, it's important that we maintain them as a vital weed control option for our agriculture. So although, as I say, I've been working on auxins in the sense that they're plant growth regulators and in their natural environment, I'm also interested in how they work for the auxins as synthetic herbicides. So this slide shows most of the auxins which have or are being used still as active weed control agents, as herbicides. Now, the one compound that's not on here is the natural hormone indole 3-acetic acid. But all these other ones have been or are used as synthetic. And you can see that there's a wide variety of chemistries which have been incorporated into the auxin fold. And I may refer to these as different scaffolds. In other words, different chemistries at the heart of each compound. So we have 2,4-D down at the bottom here, bottom left, one which I've mentioned already, and there's a whole family of phenoxyacetic acids as synthetic auxins. And then others I'll mention today will be the naphthalacetic acids. Of course, we had dicamba, we had that in our seminar last week. Dicamba up here in the benzoic acid family. And then I'll be mentioning quite a lot these pyridine derivatives and the picolinates later on in my talk. So I work on these target sites for auxins, and in particular, I'll spend a fair amount of time today talking about the receptor proteins. So I'll be talking about one called TIR1, TIR1, and the receptors are the proteins which are the target sites to which these auxins are the first site of action. So they recognize the fact that auxin is there, and they trigger all the responses that follow from that recognition process. So receptor proteins are well known, if we think now about resistance, as sites for target specific resistance, target site-based resistance. And I'll mention resistance later on in the talk. But first of all, I'll talk about how receptor proteins work, how auxins are recognized. Our work, we also look at transport proteins, because transport proteins carry the auxin in plants to where they have their activity. And there are a few families of transport proteins which are well known. There are the uptake carriers of the AUX1 family, the pins, which are efflux carriers and contribute a lot to the gradients of auxins, which set up so many of the responses we're familiar with as auxins act. And then another family of transport proteins, which I'll mention when I get towards the end of the talk. So all these are potential sites for where resistance mechanisms might occur. And then we shouldn't forget that there are also metabolizing sites, which can also confer resistance to auxins. And I'll mention those in passing, but we don't actually work on those in my group. So I've put those in italics here. So I'm going to introduce you now to the site at which the natural auxin, indole acetic acid, or IAA, binds when it arrives at its site of action. This is the protein called TIR1. And I'm going to show you a short video which shows the molecular structure of this protein. So this protein has been crystallized, and this is the crystalline structure. And there are a number of folding features which you can see are colored up here, which I don't need to go to in any detail today. But you can see it's a protein which has got a big ring of structure at the top. And what's being shown now is the small part of this big protein where auxins bind. So you can see that there's quite a deep pocket there into which the auxin will bind. And here you'll see the molecule of IAA, the natural hormone auxin, bound at the bottom of that deep pocket, sitting in amongst the residues which contribute to the recognition process, which I'm going to be talking about later. And there you can see the hole down the top. You can see it looks quite deep. And the next feature which will appear is a second protein, a co-receptor, which binds on top and locks that auxin molecule into the bottom of that deep binding pocket. And this is a peptide, but it's from a protein family called the AuxIAA proteins. And this part of those AuxIAA proteins is known as the degron. So here's a summary of that video I just showed. The overall structure is shown on the left here. And we're looking at the top. And then you see that this over here, what you see, this network is the surface structure, the electron density of the TIR1 receptor protein. This green molecule is auxin, sitting at the bottom of that pocket. You can see it's wrapped up in this network. It's in a deep pocket. And then on top of that molecule of auxin, you get a peptide binding, this orange feature here. This is the degron from that AuxIAA protein, which in the crystal structure was only a peptide because that's what was applied, and that's what we use in our research. But in nature, this is an AuxIAA protein, which is a transcriptional regulator. So that means that normally when it's not bound against auxin here, it's sitting on genes in the nucleus of plant cells and by and large restricting their activity. When auxin arrives and the degron binds on top of here, these AuxIAA proteins are activated by the enzyme, which TIR1 is a component of, and they're ubiquitinated. And ubiquitination of AuxIAA protons means that they will be very rapidly degraded after that in what's known as the proteasome. And clearly, if you degrade all your transcriptional regulators, those genes where they were sitting before auxin arrived will be free to work. They will be released from repression, and from there stem all these wonderful responses, which we know that auxin controls in the natural world. And that's everything. This one molecule auxin of IAA controls everything in the green world, from the establishment of polarity in the embryo, right through to the branching pattern and venation pattern of the tallest trees. It is a truly remarkable hormone, and no doubt that contributes to the reason as to why they're also a very effective family of selective herbicides. So we're now going to work out how we can measure the binding of such small entities, of these specific molecules, one on top of the other. So we use exactly this picture that you've got on here. We use this peptide, we buy it with a biotin on one end, and that allows us to mobilize this peptide onto chips in an instrument called an SPR machine. And I'll show you how that works in a moment. But with the degron peptide immobilized, it means that when we add auxin to the system, we can measure the binding of the TIR1 protein onto the degron. So we can measure our binding interaction. And that gives us results like this. So this is a binding assay using pure reagents. The technique is SPR. I can explain that to anyone at the end if they wish. But essentially what you see is response or binding on the y-axis against time on the x-axis. And you can see the time scale is quite short. These experiments can be quite quick. And we've got a time base of seconds here. And at this point here, we've got our peptide immobilized on an SPR chip surface. And at this point, we start adding purified receptor protein, TIR1 protein. And you can see that in the absence of IAA, there's almost nothing happens. But you add IAA and we get this wonderful binding response as the TIR1 protein binds and accumulates on the surface of the chip. At this point here, we stop adding receptor, but the buffer is still running across. So all those complexes of the degron peptide plus the receptor on the surface are still sitting there, but they start to dissociate. And so we can watch our binding reaction in real time, binding and then dissociating. And this tells us a lot about how this very fundamental biochemical binding reaction works. So we take that one step forward. We have a binding assay that works on these pure receptor systems. And just to summarize where we've got to, we are using purified receptor proteins. They're not purified from plants. We express them in a recombinant system in tissue culture. It's known as a baculovirus system, so a virus is driving expression. And then we purify the receptor proteins, TIR1 I've mentioned already. It has some sisters called Auxin F-box proteins, or AFBs, which I'll mention in a minute, which we also express and purify. And so we're using affinity purified protein on an SPR instrument. And then the different colors here start to represent the pharmacology that we're interested in. So I call it pharmacology because it's the same type of work that drug discovery companies do. And of course, it's the stuff, the work, the expertise that is brought by the big agrochemical companies like Corteva, and they have been doing it for years and years. So without going into the details of these binding curves now, you can see that if red is IAA, so our natural hormone, you can see against this protein here, it binds nicely, dissociates slowly, and we get a nice binding curve. You can get different curves for different auxins, and sometimes different receptors give you different orders. And so we can look at how well compounds work. We can accumulate the pharmacology of our synthetic auxin herbicides. And so this is what we've been doing. So we're mapping now, we're using the pharmacology techniques to map how good different molecules are as synthetic auxins. And so we do the binding assay for IAA, this is our natural hormone, indole 3-acetic acid. And then we look at every possible conjugate or variant that we can with different groups added at different sites around the core molecule. And we accumulate the binding data from all of those, and an example is shown here. So this is the series where we've got an indole in each case. Here is IAA, our natural hormone in the middle, and all our results, every experiment we run, this is our positive control. We put this as our 100%. You can see that if you shorten the side chain by 1 carbon atom to indole carboxylic acid, all the activity is lost. If you extend it by 1 carbon to give indole propionic acid, you've got 10%, 10–15% of the activity left. And we know that IPA, indole propionic acid, is a natural hormone. It does work in some systems. It accumulates in legumes, for example, but its activity is far lower than IAA. And indole butyric acid, used in some commercial purposes, but also occurring naturally, but you can see it doesn't work on the receptor. In other words, it has to be converted from the 4-carbon chain down to IAA, and this is the active compound that works when you add IBA into the system. So we have a wonderful binding assay, which is quantitative and relatively easy to do. And we can go on and accumulate binding assays for all the pharmacology that we know about for our synthetic herbicides. So this is where Corteva come into the story, because they've for a long time been developing the picolinates as a family of herbicides. And they published a paper on it, some couple of years ago, to tell the history of how they've been developing this as a scaffold, picolinic acid scaffold, for a series of successful auxin herbicides. And then Terry Walsh and colleagues there in 1999 came across derivatives where they'd added aryl groups, in other words, aromatic groups, benzene rings, onto the 6th position of picolinic acid, often with other additional electron withdrawing groups like chlorine out here. And that gave rise to a compound called DAS534, which in time they found gave rise to a family of new auxin herbicides, which are represented on the right here, and which have been launched in the last few years: Arylex acid and Rinskor acid. And so they'd done all the discovery work beforehand, but they came to us to ask them how well they bound in our system. And so we added these into the binding assays that I've been describing. So here I introduce on the left the family of sisters to TIR1. So all plants, all higher plants, contain a set of these receptor proteins like TIR1. This is the family from Arabidopsis, and most other plants actually do contain multiples of 3, 6 is quite common, sometimes up to 7 with another branch. So we have the canonical or parental, the stem type here, TIR1, and it has, due to gene duplication, a very close sister, AFB1. More distantly related is AFB2 and 3 as a pair, and then more distantly again, you get round to AFB4 and 5. And each of these ones in the ring, we've expressed in our system, and we've done these binding assays using our SPR system. And you see a set of results, the pharmacology results on the right. So in all cases, the color code is the same. So the red graph is for the natural auxin IAA, and against TIR1, the one which was crystallized, the one I've shown you the structure for, you can see that IA gives a strong, steady binding response. And as I've shown you before, that complex, once made, is fairly stable. It dissociates, it falls apart relatively slowly. And that contributes to a high affinity interaction. When you look at some of these other auxins, these other scaffolds, 1-NAA gives a very similar, slightly weaker, but very similar response to IAA. 2,4-D, our long-standing herbicide of the family of phenoxyacetic acid, it's got reasonable binding, but notice here that when you start to dissociate the complex is far less stable. It falls apart much more rapidly. And that's why this does not reach the same amplitude of binding as, for example, IAA, because all this time that it's binding here, it's also falling off. It's falling apart much more quickly. So it's not going to reach this same amplitude up here because that's a fast off rate. And then we've got one of our new 6-aryl picolinates. We've got Arylex acid here in blue, slightly weaker response again, and then quinclorac at the bottom, a very weak response for quinclorac. So those are the binding responses against our canonical receptor TIR1. AFB2, the next nearest neighbor in this family, you can see that the responses are quite similar. Certainly the order of specificity is the same, but you can see here that perhaps binding is a little more rapid, but in all cases, the dissociation is also more rapid. The complexes are slightly less stable. So we have the same preferential selection for different auxins, but this one will have slightly lower affinity. Now it becomes more interesting when we deal with AFB5. So this, again, slightly more distant family to TIR1. So against this receptor family AFB5, notice the scale has been expanded on the y-axis here. We still have IAA, the natural hormone, in red. And as ever, we're representing that binding at the end of the association phase as 100%. But now this new auxin, and indeed all the 6-aryl picolinates, is binding far, far more strongly than even IAA does to this receptor class. Beyond that, again, NAA is doing much the same as it has with the others. It's slightly weaker than IAA, but shows the same pattern. Likewise, 2,4-D. And a correction against one identification we've published before is that quinclorac does not bind well at all to AFP5, as it binds pretty poorly to all these receptors in this family of receptors. So we have a class of auxins, synthetic auxins now, which are binding preferentially to AFP5 and relatively weakly to TIR1 and AFP2. So that's very interesting. And of course, as I say, Quantitative have taken the best path forward with those and have registered them. And some of them have turned into active herbicides. And this is one of their pictures of one of their trials of the Rinskor version, Rinskor active, which is a formulation of Rinskor acid as a conjugate. And it's applied to a rice field here. Rinskor tends to be used in rice at a very respectable low field rate, and you can see that it's given very respectable control of the weeds against the untreated plot here. So a successful collaboration to explain a slightly different route to action and specificity, and distinguishing it from 2,4-D and some other herbicides in the way that it's acting, or the route through which it's acting, perhaps. So just as we've done for the 6-aryl picolinates, of course, we have a binding assay that we've used for many, many compounds. And this is a whole series of compounds, and it somewhat repeats what I've said already. In these two plots, we've got TIR1 at the top and the least similar one, AFP5, at the bottom. In both cases, we've got IAA on the left and at 100%. And the bottom line here really is that almost nothing is as good as or stronger. Very few things are stronger, significantly stronger than IAA for TIR1. But for AFP5, both the pattern of activity and indeed the strength of activity is very different. So auxin receptors are selective and different, perfect for pharmacological type approaches. But of course, these histograms mean very little if you're trying to design new molecules. So here we turn to the computational chemistry resources that our friends in what we consider the real pharmacology companies, although it's the same science, the drug discovery companies have helped develop. So this is bits of software which allow you to amalgamate the data we have from binding with the knowledge that we have of the molecules themselves. So that generates what we call a pharmacophore. In other words, the best possible molecular shape for an auxin to be if it's going to be active. And it's easiest to explain in terms of molecular shape how it works. So we have binding activity for over 400 auxin and auxin-like compounds. We know whether they're good or bad, and we can quantify that. You put all these compounds into the software as molecules, and the software calculates features like the molecular shape in 3 dimensions for each compound, where hydrophobicity lies, and where local charge on the molecules lie. And then it uses some elaborate statistics to create from your data the weighted average of the best average auxin. And on the right, you see what that is here. Superimposed is the natural auxin IAA, and you can see it fits in this shape beautifully, as you'd expect. But there's some wriggle room. There's space here, perhaps, that you can exploit, which will allow a compound to be an active auxin. That's where these other features come into play, because we know that this ring system is aromatic. In other words, it's hydrophobic. It prefers oiliness to wateriness. And instead of showing the average hydrophobicity on the molecule, or the hydrophobic shape as we could have done, what we've shown is a color scale for where if you add hydrophobicity, it's green. And if it's green, it means it improves the activity of that compound. So if you add a hydrophobic group into this space here, for which there's space in the shape, or indeed over here, you improve the activity of your molecule. In contrast, if you were to put a hydrophobic group which fills into this magenta space, you would ruin the activity of that molecule. And so you can see now that we've built up effectively a computer-based set of rules about what is an auxin, or what can be an auxin, and what can't be an auxin. And you can use this either to in silico screen for molecules, or to explain the activity of molecules. And I'll go forward and play this as a movie. This is the pharmacophore for TIR1. That's IAA in it again, and you can see you can turn that round in 3 dimensions, and you can see exactly, again, this is the hydrophobicity cliff, as it's called over here. If you add hydrophobicity into this green area at this side of the molecule, you'll improve its activity. And so you can get inside these molecules and start to design or select compounds which are of interest to you. And to take that one step further, of course, we can put these pharmacophoric models inside the receptor, for which I showed you the crystal structure at the beginning. And here you have the average hydrophobic space occupied by auxins in the middle of the amino acids which make up the receptor binding site inside TIR1. And we see the same thing from the top on the left and from the side over here on the right. And this helps to explain why auxin binds so tightly, so selectively into this binding site. And then if you go down to the bottom here, again, you see these design cliffs, and you can see that if you add a hydrophobic or electron withdrawing group over in this green cloud, that extra hydrophobicity will sit between these two phenyl acetic acid residues of the receptor. And that explains why hydrophobicity is allowed in this area and will help improve activity, because those go together well. So we can use these models to help us with new chemistries. So if I just summarize this part of the talk, we have a good binding assay, and we can do structure activity relationship studies. And these have been done to give detailed 3-dimensional maps of what makes the perfect auxin. They will be dependent, of course, on each receptor, and we have them for each receptor class. So we have them for AFP5 as well as TIR1 and so on. On the way, we've observed and mapped the fact that the novel 6-aryl picolinates have a strong preference for AFP5. And I haven't mentioned it today, but we've presented a molecule, an indole-based molecule, which has a strong preference for TIR1 and doesn't like AFP5, so the opposite selectivity, if you like. So let's now look, now we know how auxin binds, how it's recognized, we need to think about resistance. So what might happen to any of these systems which would confer resistance into the auxin response mechanisms of plants? So I'm going to talk later about target site resistance. I've talked about the target site itself. I'm going to come back to that at the end. But on the way, I'm going to tell you a little bit about some of our work on auxin transport proteins, and on I'll touch on metabolism, just from the perspective of the molecular biochemist. So this very simply is the plant's auxin transport system. So here is a plant cell, very simply. And we know that auxin is the one hormone which has polar transport in a plant. So there's a net direction of movement of auxins. That's facilitated by these uptake carriers, which I've mentioned, the Aux family, the Aux1 family, Aux and like Aux1, the lax proteins. And these carry auxin into cells. At the other end, and these tend to be the ones which dominate on the directionality of flow, are the so-called PIN proteins. And these are the efflux carriers. So they're pumping auxin out of cells, and that gives directionality to gradients. In addition, there is another family of exporters known as the ABCB transporters. And these are familiar to any eukaryotic cell type. Indeed, prokaryotes, microbes have this type of transporter as well. And they're involved in the transport of all sorts of small molecules and many xenobiotics. So when we're thinking about resistance, clearly, if we have a mutation or something which affects AUX1 so that it reduces uptake of auxins, you might confer tolerance or resistance. Similarly, if you increase efflux, as long as it's carrying your xenobiotic, the synthetic auxin, if you increase efflux, you will have a more tolerant, a more resistant cell, a more resistant plant to those hormones. And because of that, we've started some work to look also at the pharmacology of these transporter proteins. And we've done the same as I've described for TIR1 to the uptake carrier, the AUX1s. And here is that pharmacophoric model for AUX1. And you can see in this case 2,4-D, which is a very, very good substrate for the uptake carrier. 2,4-D is taken up into plant cells very effectively by these transporters. And you can see it fits beautifully inside the pharmacophoric average best shape for uptake. And indole 3-acetic acid fits in here as well. It would be across here. But when you look at the two side by side, AUX1 against TIR1, you can actually see that there's a much tighter fit. In other words, the transport proteins are more selective than the receptor in terms of what they will carry, which is a bit surprising, but perhaps not too surprising. But to add to that, we did find that several auxin herbicides are not substrates for the uptake carrier. So although this carries 2,4-D, it will not, for example, carry dicamba or picloram or quinclorac. So they get into cells and work perfectly well as herbicides without having to go through this uptake system. There are other transporters which will carry them into the cell. Therefore, the uptake carrier is not necessary for herbicidal activity. And although mutants in this protein AUX1 will confer tolerance to 2,4-D, it's not likely to be a big problem in terms of field resistance to auxin herbicides. If we now turn our attention to the other end of the cell, we are working on PIN proteins, but we have no pharmacophoric data. These are much more difficult proteins to work on. But ultimately, we will hope to present the same sorts of models for PINs, and we're not working on ABCBs. But the thing to say about ABCB proteins, this class of transporter protein, is, as I mentioned, that they are common to most life forms, and the large part of their purpose in microbes is to export xenobiotics. And it's this class of proteins which confers resistance in, for example, the multiple resistance bacteria like Staphylococcus aureus, which cause clinical problems when you have multi-drug resistance to these microbes. So we can consider these as very likely sources of resistance, not through mutation in this case, usually through upregulation. But we might, as it says in the note here, we might expect each of these effects to be compound specific, just as they are for the uptake carrier. So not necessarily all synthetic auxins will be susceptible in the same way. And now just one or two slides on metabolism. So again, metabolism will be dealt with by others, I know, but metabolism of an active compound is clearly one way of removing it from the system, from the living organism, so that it is no longer toxic. And there are some familiar chemistries or biochemistries to do that. And we do have some crystal structures of enzymes involved in these processes. So here is the crystal structure of one of these proteins called, named as GH3. These are enzymes which add an amino acid group onto IAA in the natural situation. And by making the amino acid conjugate of IAA, it renders it inactive, and it will be transported almost certainly into the ER or into the vacuole, where it will be harmless. There's no reason to believe, and indeed there is evidence to show, that GH3s will metabolize, for example, 2,4-D in exactly the same way, and so could confer resistance. on the right here, we see the crystal structure of a class of enzymes known as the GSTs, or glutathione S transferases, GSTs, glutathione S transferases. And those that have been following Rob Edwards, Paul Neve, and the Blackgrass collectives in the UK on the mechanisms through which resistance to a wide range of herbicides is conferred. It's through upregulation of this family of enzymes, the GSTs, or a class of this family of enzymes. And here in this GST, you can see an indole. It's not exactly an auxin, but it's an indole. And these enzymes add glutathione onto the xenobiotic, into the small molecule. And again, that glutathione conjugate will be transported very rapidly into the vacuole where it will be harmless, a tolerance or resistance mechanism. And that upregulation of GST, so the accumulation of GST enzymes, is a very well-known resistance mechanism. And that could occur for auxins just as it does for other classes of herbicide. But I might say that for each different scaffold of auxin, probably it will take a different class, a different member of each class of enzymes to metabolize them. Not necessarily, but quite likely. So cross-auxin resistance is less likely with these mechanisms than it is through target site resistance, which I'll come to in a minute. And we heard about the widespread use of dicamba and some of its problems last week. Of course, some of our seed companies have developed genetically modified crops which are tolerant to dicamba and to the phenoxious auxins as a mechanism of managing weeds in fields. And these make use of microbial enzymes, which in both cases actually oxidize the molecule to deactivate it. And this is a very good synthetic way of rendering crops resistant to these molecules and making them widely available. But it just goes to show that metabolism is possibly a very effective way of rendering resistance in crops. And of course could arise in weeds. One last observation here, and this is a bit of work done with Paul and actually with Dr. Lee from Nanjing Agricultural University, and that is that we tried to look at whether or not resistance to one auxin was metabolic resistance was also going to be carried across to other classes of auxins. And very quickly, this is a genome-wide association map, and it shows where a position on a chromosome where there is a locus for resistance to, in this case, dicamba. And you can see that although there is a metabolic resistance to dicamba occurring at this locus in Arabidopsis, which is the populations that we use for that, the same doesn't hold for fluroxypyr or some of these other auxins. And so the chance of cross-resistance between auxins from metabolic resistance mechanisms is far less likely, which is very good news as we try to manage auxins as a family for continual use in food production. And then as the last word on this very slide, this slide says, there's a but. That's metabolic resistance, and I'll summarize this part of the talk. There is scope for resistance to arise to auxin herbicides through a range of familiar biochemistries, but by and large they haven't for one reason or another, or we haven't yet been able to map them as closely as we'd like, and that's to do with the genetics of these weeds that we deal with. Transport and metabolism seem less likely to confer cross-resistance to a set of auxins than target site resistance at the receptor complex, because here we're dealing with the core binding event, which clearly the receptors recognize all different types of auxins because that's their purpose. And this is where I refer now to someone else's work, which is the paper published by Leclerc et al. In PNAS a couple of years ago. Cross-resistance to dicamba, 2,4-D and fluroxypyr in kochia is endowed by mutation in an AUXIAA gene. Now, if you remember back to the binding assays, the degrons that we were using in our binding assay derive from AUXIAA proteins, which are our transcriptional regulators for auxin. So right down at the receptor complex here. There's a mutation at the receptor complex, but it's not in TIR1. It's not in what I've called the receptor itself. It's in this co-receptor, the AuxIAA protein. Here is some of their evidence, and here's the dicamba evidence up here. They're using root length or the inhibition of root length as an assay. And you can see that the sensitive kochia is inhibited at far lower doses than the resistant kochia. And you can see that resistance is conferred also for 2,4-D, but perhaps not for some other auxins down here. So they were very lucky to be able to map. It's one of the few examples for auxins where the resistance has actually been mapped right down to the gene that's responsible, and they mapped it down to this AuxIAA number 16. This is a structure of the gene, and at the very heart of the protein when it's translated, as I've mentioned, is the degron. This is what binds in our binding assay, what binds in nature in the crystal structure of the receptor. And that degron has a core repeating motif sequence of GWPPV. WPP are what sit right on top of auxin in the binding site. And you can see that in the resistant genotype, this glycine, this G at the beginning of the degron motif, has been mutated to an N, an asparagine, which in terms of amino acid similarities is a big, big change. And there's a set of sequences put on the figure down here just to show that most AuxIAAs have GWPPV, including the wild type sensitive kochia IAA16. And then here is the mutant version in the resistant form. So how is this working? How is this not working? So we now go to a set of experiments that we did a while ago. We've not been able to do the binding assay with the kochia sequence for IA16. We just never started that. We never looked at that. But what we have done is we've looked at another series of mutants, which are long-standing in the literature. They relate back long before we knew about receptors, to a set of mutants identified by Otto Liese and company in 1996. And these are the AuxIAA proteins known as AuxIAA17. Here in the box is the degron sequence. In the wild type, you've got GWPPV, just as I've shown you for the sensitive version of IA16 in kochia. But these mutants they identified have got mutations within the degron. So in 17.1, the second P is replaced by an L. And in 17.3, the final V is replaced by a glycine, a G. And if you then look at how these bind compared to the wild type, following the same color sequence, the light blue here is what we're familiar with for TIR1. In the presence of IAA, binds strongly, fairly stable complex. If we go to 17.1, where that core degron has been mutated, the dark blue down here, you can see that there's almost no binding at all, even in the presence of auxin. So this mutation at the heart of the degron has prevented the binding, the assembly of this co-receptor complex. If you take one step outside the core and go to this G, what you get is this intermediary phenotype with the binding here. So it binds quite strongly, quite respectably, but then once assembled, you can see that that complex is very weak. It falls apart easily, rapidly. So where the residue, the mutation is just outside the core of WPP, the binding happens, but it's weak. And that's what I'd anticipate would happen with this kochia resistance here. Although it's at the other end of the degron, I would anticipate that there's a binding, but it's a weak binding. And if we relate that to the phenotypes, which were published in this paper in 1996, you can see here the perfectly respectable wild type IAA17 plant, the nice Arabidopsis rosette and perfectly happy flowering stems coming up here. The severe 17.1 mutant is a poor weedy plant. And then the intermediary binder, which binds but then falls apart, is an intermediary phenotype. So the phenotypes correspond very nicely with what the binding data are telling us from our laboratory experiments about the binding in the degron. So there's a nice correlation of activities there. And I anticipate that the same, as I say, is true for this resistance mechanism here. So the nice thing as part of this paper, and I haven't shown you the data, but that these mutants, these IA17 mutants, are also resistant to 2,4-D. There's a very significant shift in dose response for both of these to 2,4-D. In other words, they're 2,4-D tolerant, 2,4-D resistant. If we now turn that around, so we've got exactly the same set here as we have with kochia, if you turn it around, what you can also see is that where there's no challenge, the phenotype is pathetic. In other words, there's a huge fitness cost to these mutations. So it might be under the selective pressure of herbicide presence, this plant on the left might survive and shed a few seeds, be resistant to 2,4-D, but in a wild type situation in a population, it would soon be excluded. It will soon die out from the population. It will be selected against. And that was again shown in this very nice paper by Napier et al. And although these numbers are a little bit small perhaps to see, you can see over here that in the homozygous resistant, the homozygous sensitive, and then the heterozygote in the middle here, that when you look at, for example, biomass, there's a significant penalty in the resistance one when it's grown in competition, less so in monoculture. So a very nice example of resistance equating to a loss of fitness when there isn't the selective pressure. But that resistance is very definitely at the heart of the recognition, the core recognition receptor complex for auxins. So if I summarize this last part of the talk, resistance has arisen to auxin herbicides in wild populations. That's undoubtedly true, not as frequently as it has to other herbicide collections, but it has happened. One clear case of target site resistance has been reported, which is the Napier paper in kochia, which confers resistance to a number of different auxin scaffolds. And here an advert for Todd Gaines' seminar later in the series, tomorrow I think, and he'll tell you about another very similar but different example of, again, the co-receptor being responsible as the site at which resistance is conferred. But no more details of that from me. But the fitness cost of this resistance suggests it could be managed with good stewardship of our herbicides. So when I first gave a version of this talk to the Global Herbicide Resistance Challenge meeting in Denver in 2017, these were some of the closing statements that I made. Receptors are redundant. In other words, there's a family of them which all work the same way, the TIR1 and AFBs. And so I said that mutation of any one is not likely to lead to resistance because the others would still be there to work. But care should be taken on managing the picolinates because there was so much selectivity for AFB5. I have to now, of course, update that. So this is the synthesis today. Receptors are redundant. And I've said, so mutation of any one is not likely to lead to resistance. Well, that's true if you talk about TIR1, but it's a co-receptor complex. So there's two contributors to it. And the bit that I'd overlooked in 2017 has very soon afterwards shown me to be, to have overlooked a very important player, and that's the co-receptor, these AuxIA proteins. So mutations in co-receptors are proven sites for resistance in field populations of weeds to our auxin herbicides, and this is cross-resistance. However, there is a fitness penalty. So just to finish on these other points and things that I've covered, the transport proteins are potential sites for resistance, but mutations in AUX1, whilst they can confer tolerance in the lab, it's not likely that that will be a big problem in the field until someone comes along again and tells me that I'm wrong on this. Upregulation of efflux proteins, whether they're PINs or ABCBs, could easily confer resistance, but we haven't known examples of these yet. And then, of course, everything could happen with metabolizing enzymes. In general, this is not due to mutation, it's due to upregulation of metabolism, which naturally gets rid of xenobiotics. But these may well be, in fact, I might say, will be likely to be scaffold specific. So in other words, if it deals with 2,4-D, it probably won't metabolize dicamba, for example, or the aryl picolinates. And that's helpful for how we think about managing our cohort of auxin herbicides. And so a very last slide. My work on the chemical biology and pharmacology of auxins continues to give new insights into one of the most important events in plant biology. So outside our herbicides, of course, auxin is key to plant life. And it recognizes the role of this vital hormone and how it's selectively recognized by plants. But the work also helps us understand auxins as herbicides and the molecular basis of resistance. In the final picture, some of my colleagues who helped me with this work and the funders. So I thank you for listening now, and pass back to Louis, who will manage the questions.
Thank you, Richard. It's very good to see your work, and I was there in Denver when you first present here. This presentation is very good to see all the information you have on the Auxin herbicide, and I'm pretty sure that in the next few years you're going to have lots of new information on that.
Let's hope.
Yeah, we have several questions here. I will read to you some of those, and some of them are already answered during the presentation, so I won't read it. So let's start with this one. Juan Camilo is asking, how is quinclorac mode of action if is not the same receptor as the receptor you showed?
Now, that's a very good question. And the honest answer is I don't know the full answer on this. So what our data there showed is that there was a very low level of binding. So this doesn't mean that it doesn't bind, it just means that it binds fairly poorly. Now, the thing about these synthetic compounds, of course, is that if there isn't a very active metabolism, and due to various accumulation measures, they often accumulate inside cells to quite high concentrations. So they're accumulated at concentrations that are higher than they're applied at. And there are papers published that show this. So they accumulate in cells, and so the concentration builds up very high, and therefore what appears as a low, weak binding event when we do it in vitro, in vivo, it could account to something which is significant, and of course it would be long lasting because they wouldn't be metabolized. So the signal would persist much longer than a natural signal would do. But it's quite possible that there are other sites at which quinclorac works as well. And of course, I don't know what those might be.
Another question from Thiago Gazola. Is there any difference in mode of action of 2,4-D and dicamba? Maybe at gene level?
So in terms of the binding response, both of them are less strong. I mean, I showed the data for 2,4-D. Dicamba falls down towards a slightly weaker binding interaction as well, but would again accumulate and persist, just like I've been describing for quinclorac. And it will bind in the same site. It's a small molecule. So the result in terms of the natural biology will be the same. Having said that, there is an old paper from the Jones group which looked at transcriptomics, which showed that each different auxin had generated a slightly different genetic signal in whole plant sampling. So there may well be local or cell type specific events because of the particular receptor co-receptor complexes which occur in those cells, which are activated more or less strongly by dicamba, which would give some selectivity. But in terms of a herbicide, the outcome is the same. The overdose will kill the plant.
Mm-hmm. Another question here is regarding selectivity. The selectivity of grasses could be due to both auxin affinity and TIR1 selectivity?
Yes. So, I mean, of course, the beauty of a selective herbicide, the auxins as selective herbicides, is that they can be used by and large on cereals to kill the broadleaf weeds, and it's long been a question as to how we explain that selectivity. And there's a sort of tacit understanding that it's to do with improved, better metabolism in the grass or the cereal. But I think that's an open question. We don't know. In terms of the receptors, we have actually looked, we've looked with Corteva at TIR1 receptors from grasses and from Arabidopsis and other dicots. And we're not quite ready to release the answers on that yet. Sorry.
There is another question from Dr. Anderson Nunes. My view on your data presented is that it may have a synergetic, show that yes, there is a synergetic activity of mixture of auxin herbicide in most cases. Do we agree on that? Yeah.
Yes, I mean, I think that's quite a good rule of thumb, is that you mix your mechanisms of action. Although this is the same mechanism of action, but it would be a promising way to help manage resistance, or the resistance arising if you did mix them. You'd still need to think of using other mechanisms of actions to make sure that you're not just constantly selecting, because you could select for another, Auxin IA mutation, which would be resistant across the auxin stable, and that would be a problem.
Mm-hmm. Yeah, there is another question from Aldo Meroto. He's coming back to the quinclorac activity. There is just a few plants affected by quinclorac, contrasting with the large number of plants affected by 2,4-D as an example. Is that more related to different receptor or metabolism between the two compounds?
Again, I can't fully answer that question because I've never looked at the metabolism of these compounds. I'm sure that the manufacturers have looked at metabolism as part of registration processes and things, but I don't know the answers to those. So an educated opinion, if you like, is that a lot of it's to do with uptake as well as metabolism. So the accessibility of the compounds to get into plants and into plant cells. So that's the basis of some selectivity or some failure to act in some cases. But in large part, I suspect it's to do with different metabolism.
Mm-hmm. What would be the most important environmental factor affecting auxin in plants? I understand that's auxin herbicide, maybe.
Sorry, say that one again. What's the most probable?
The most important environmental factor affecting auxin in plants.
The most important factor in terms of the molecule is that clearly that it's recognized by that receptor complex, and to be really active, it needs to maintain a nice stable receptor co-receptor complex, because that will activate the auxin response system most effectively and for as long as possible.
Mm-hmm.
I'm not sure I've answered that well. If you can think of another way of following that up, please do.
All right. Yeah, it was not clear if he was asking about the herbicide or the auxin itself, how it acts.
So the core of action in all these cases is exactly the same, that the genes the auxin responsive genes are activated as a response of the breakdown of those transcriptional regulators.
There is several that ask similar questions, so I will try to wrap up in the same question. What's the probability? I think you answered that during your presentation, but only to wrap up the information. What's the probability of selecting weeds resistant to cross resistance to other auxin herbicide?
Well, we know of two the one that I mentioned, and of course Todd will mention another one. Two occurrences where field resistance, which is cross-auxin, has occurred against all the weeds that that we have as challenges across the world. So the probability is incredibly low, but of course it does arise, and these resistances do tend to spread around a bit if it's not managed well. So if the question is about how important a problem it will become, I think probably you as weed scientists are better placed to answer that than me as a biochemist. Yeah, I'll pass it back to you, Louis.
Yeah, yeah, that's a quick, difficult question to answer. I think in the future we'll see the real answer on that, but that is likely to have a cross resistance for sure. But in different, like the metabolism is one of the most likely to have cross resistance or promote cross resistance. Considering the picolinates has high affinity to AFB5, the possibility to have a cross resistance to another molecule such as 2,4-D and dicamba should be less likely, more probably to non-target site, or do you see any possibility for a target site?
So I think that so if the aryl picolinates get used so heavily, so widely, and without any without wise stewardship, and they are being used with wise stewardship, so hopefully this won't arise. If you get resistance to the aryl picolinates, because they're so selective for AFB5, there's a very good chance that those plants will remain susceptible to 2,4-D and dicamba, which are working far more strongly through other members of the receptor family. But that only that's probably the only case where that might work. Yeah, I think that addresses the problem.
I think we're gonna have the last questions. The last question. The ABP1 protein is no longer a receptor.
So there's a very good question, because I did spend a lot of time earlier in my career working on ABP1. I mean, the paper published in, what was it, PNAS, I think, about 5 years ago now, showed very convincingly that if you knock ABP1 out, because there's only one member of that family, there's only one gene for ABP1, an intact ABP1. If you knock it out, there was no particular phenotype, no gross phenotype anyway. And that suggested straight away that in fact it was not working as a receptor. And after that, there were a number of publications which explained effectively the false positive results that had been gained through a number of the tools which had been developed to explore the activity, the function of ABP1. And so at the moment, although I know people are still working on it, and it's not a closed story yet, but it seems unlikely that ABP1 is a target for Auxin herbicides, and that I think we can probably consider that it's not a receptor in the same magnitude, of the same importance that TIR1 and the AFBs are. So it's not an absolute no, but essentially it's, to all functional purposes, it's a no. It's not a receptor.
All right. We have so many questions. Your presentation brought us a very good insight on Auxin herbicide mode of action and resistance. Thank you very much for your excellent webinar that you shared with us. It was excellent. Before we finish, we would like to invite you to participate in our next webinar. It's going to be tomorrow. Dr. Todd Gaines. He will speak about the novel herbicide resistant mechanism for synthetic auxins. And on June 25th, Dr. Cristiano Piasecki will speak about application of omics and synthetic biology towards the elucidating of non-target site glyphosate resistance mechanism on kinase. Dr. Napier, thank you very much. It was excellent. Everybody for sure is more educated about auxin herbicide mode of action. Thank you very much for your willingness to be here and for your time.
My pleasure. Thank you very much, everyone.
Thank you. Bye-bye.
Bye.